lecture which we are studying about lipolysis. In that lecture, we got to know how the triglycerol will be separated and a glycerol bone will pass towards the liver and the fatty acids will transfer towards the mitochondria. So we're gonna know what happens to those fatty acids in mitochondria today. So let's see. In mitochondria, there is an enzyme which will let the fatty acids inside by marking. It is known as acyl transferase. So this enzyme is responsible for allowing the fatty acids inside the mitochondria. So after once the fatty acids will enter into the inner layer of mitochondria, there is an enzyme which is known for carnitine acyltransferase. It will recognize this fatty acid which was led by acyltransferase and it will let it pass towards to the inner layer and at there is another type of carnitine acyltransferase which will allow it to proceed towards the beta oxidation. In beta oxidation, there are three products will be evolved, which are known as NADH plus H plus. Before we go to NADH and H plus, we have to remember 2 points. Vitamin B3 is known as NAD and vitamin B2 is known as FAD. Whenever the hydrogen atom form a chemical reaction with B3, it will form NADH plus H plus. And whenever hydrogen atom makes a chemical reaction with B2, it will form FADH2. So in the beta oxidation, NADH plus H plus will be evolved and FADH2 will be evolved and along with them acetyl CoA will be evolved. So when we check how many ATP will evolve from one fatty acid, we'll get to know one NADH plus H plus atom is equal to 3 ATPs and one FADH2 is equal to 2 ATPs and one acetyl CoA is equal to 12 ATP, by which we will understand that one fatty acid can make 17 ATPs. And after acetyl CoA evolves, it has to undergo towards the Krebs cycle to form remaining 12 ATPs, which we're going to look at now. So this acetyl CoA will enter the Krebs cycle. There is an enzyme which is known as pyruvate dehydrogenase, which will give permission to enter into the Krebs cycle. Krebs cycle is a cyclic reaction. It will be a cyclic reaction. So once the acetyl CoA enters, it will mix with oxaloacetate and form 6 carbon atoms. Oxaloacetate has 4 carbon atoms and acetyl CoA has 2 carbon atoms. Together they make citrate, which will make them 6 carbon atoms. So once they become as a citrate, they will convert into isocitrate. At isocitrate, we have isocitrate dehydrogenase enzyme, which will break one carbon atom. Whenever the carbon atom breaks, hydrogen will be released. And whenever the hydrogen will release, vitamin B will be present there to capture it. So once the carbon atom will be released at the isocitrate stage, there is vitamin B3 to capture it and it will make NADH plus and it will send it towards the storage to make ATP. And after the isocitrate stage, there is a stage called alpha ketoglutarate, and once the they become as a alpha ketoglutarate, there is another enzyme which is known as alpha keto dehydrogenase, and again it will break one more carbon atom, and hydrogen will be evolved, and vitamin B3 will be there to present to capture it. And once it captures, it will be sent towards storage as NADH plus H plus. And after the stage, there is cycline CoA. Sorry, succinyl CoA. There is a stage called succinyl CoA, and once the which will have only 4 atoms, 4 carbon atoms. So in the succinyl CoA stage, there is already existing GDP and one inorganic phosphate, so will be transferred towards them by process which we already know, phosphorylation, and they form GTP. One GTP is equal to one ATP. So after this succinyl CoA stage, it will become as a succinate. In succinate, from succinate to formate, fumarate, there is another beta atom which will be present to capture the hydrogen which is present in the existing 4 carbon atoms, and it captured and it forms FADH2, and it will be transferred to the storage. And after the fumarate stage, we have a malate stage, and in the malate stage, again there is one more carbon atom, one more hydrogen atom which will be existing in the 4 carbon atoms, and it will be captured by B3, and it forms NADH plus H3. So total, when we calculate, we're going to calculate how many ATPs are formed now. So one ATP is already formed because of one GTP, and there is a NADH plus H plus which is formed at isoseparate stage. So total it will become 3 plus 4. 3 plus 1 is equal to 4, and after that there is one more NADH plus H plus. Now it becomes 7, and there is one FADH plus H2. It will become 9, and there is one more NADH plus H plus. Now they become 12. So hence proved 12 ATPs will be evolved in the Krebs cycle. So this is all about the Krebs cycle.